Welcome to our exploration of analytical tools for biological networks. In the world of biology, networks are everywhere, from the intricate web of protein interactions inside a cell to the vast connections between species in an ecosystem. Understanding these networks is crucial for unraveling the mysteries of life. Today, we'll embark on a journey through the essential analytical tools that scientists use to decode these complex systems. Whether you're a student, researcher, or just curious about how life's connections are studied, this video will guide you through the basics and beyond. Let's start by understanding what biological networks are and why they matter. Biological networks are representations of relationship between biological entities. These entities can be genes, proteins, metabolites, or even entire organisms. The connections or edges between them represent interactions such as binding, regulation, or communication. By mapping these networks, scientists can visualize and analyze the complex interplay that drives biological processes. For example, a protein-protein interaction network shows how proteins work together to carry out cellular functions. Understanding these networks helps researchers identify key players in health and disease and can even reveal new drug targets. There are several types of biological networks, each with its own focus. Gene regulatory networks map how genes control each other's expression. Protein-protein interaction networks show how proteins physically interact. Metabolic networks trace the flow of metabolites through biochemical pathways. Ecological networks, on the other hand, illustrate relationships between species in an environment. Each type provides unique insights, and the choice of network depends on the biological question at hand. By distinguishing between these networks, researchers can tailor their analytical approach for maximum impact. Analyzing biological networks allows scientists to uncover hidden patterns and relationships that are not obvious from studying individual components. For instance, network analysis can reveal which genes are central to a disease pathway, or which proteins are most influential in a cellular process. This holistic perspective is essential for systems biology, where the goal is to understand how parts work together to create the behavior of the whole system. By leveraging network analysis, researchers can make discoveries that would be impossible with traditional reductionist approaches. Before analysis can begin, data must be collected. Biological network data comes from a variety of sources, including high-throughput experiments like DNA sequencing, proteomics, and metabolomics. Public databases such as String, BioGrid, and KEGG provide curated interaction data. Researchers also use literature mining to extract relationships from scientific publications. The quality and completeness of the data are critical, as errors or gaps can lead to misleading conclusions. Careful data collection and validation are the foundation of reliable network analysis. Once data is gathered, the next step is constructing the network. This involves defining the nodes, such as genes or proteins, and the edges which represent their interactions. Construction can be straightforward, like mapping known protein interactions, or more complex, such as inferring regulatory relationships from gene expression data. The resulting network is often visualized as a graph, with nodes connected by lines or arrows. Proper construction ensures that the network accurately reflects the biological system under study. Visualization is a key part of network analysis, helping researchers see patterns and relationships at a glance. Several software tools are available for this purpose. Cytoscape is one of the most popular, offering powerful features for visualizing and analyzing complex networks. Jephi and Pagic are also widely used, each with unique strengths. Good visualization makes it easier to communicate findings and spot trends that might otherwise go unnoticed. Cytoscape is a free, open-source platform designed specifically for visualizing complex networks. It supports a wide range of data formats and offers extensive customization options. Users can import network data, apply different layouts, and use plugins to extend functionality. Cytoscape also integrates with databases, making it easy to update networks with the latest information. Its user-friendly interface and powerful features make it a favorite among biologists and data scientists alike. Jephi and Pagic are alternative tools for network visualization and analysis. Jephi is known for its interactive interface and real-time visualization capabilities, making it ideal for exploring large networks. Pagic, on the other hand, is optimized for very large networks and offers advanced analysis features. Both tools support a variety of file formats and can handle networks from different domains, not just biology. Choosing the right tool depends on the size and complexity of your network, as well as your analysis needs. After constructing and visualizing a network, the next step is analysis. Basic network analysis involves calculating properties like degree, which measures how many connections a node has. Centrality metrics such as betweenness and closeness help identify important nodes that play key roles in the network. These metrics provide insights into the structure and function of the network, guiding further investigation and hypothesis generation. Degree centrality is one of the simplest yet most informative network metrics. It counts the number of direct connections a node has. In biological networks, nodes with high degree centrality may represent hub genes or proteins that are critical for network stability. Identifying these hubs can highlight potential targets for therapeutic intervention or further study. Degree centrality is easy to calculate and provides a quick overview of network structure. Betweenness centrality measures how often a node appears on the shortest paths between other nodes. Nodes with high betweenness act as bridges, controlling the flow of information through the network. In biological systems, these nodes may represent key regulators or bottlenecks. Understanding betweenness centrality helps researchers identify critical points for intervention, such as drug targets or control points in metabolic pathways. Closeness centrality reflects how close a node is to all other nodes in the network. Nodes with high closeness can quickly interact with the rest of the network, making them efficient communicators. In biological networks, such nodes might be involved in rapid signal transmission or coordination of complex processes. Calculating closeness centrality provides insights into the efficiency and robustness of biological systems. Network motifs are recurring, small patterns of interconnections found within a network. These motifs often represent fundamental building blocks of biological systems, such as feedback loops or feed-forward circuits. Identifying motifs helps researchers understand the underlying principles that govern network behavior. Specialized tools can automatically detect motifs, revealing hidden regularities that might be missed by other analyses. Community detection algorithms identify groups of nodes that are more densely connected to each other than to the rest of the network. In biological networks, these communities might correspond to functional modules, such as protein complexes or metabolic pathways. Detecting communities helps researchers break down complex networks into manageable parts, making it easier to interpret their structure and function. Pathway analysis focuses on tracing specific routes through a network, such as metabolic or signaling pathways. By mapping these paths, researchers can understand how signals or metabolites flow through a system. Pathway analysis is especially useful for identifying key steps in disease processes or drug action. Many tools offer pathway enrichment analysis, highlighting which pathways are most relevant to a particular condition or dataset. Enrichment analysis tools help researchers determine whether certain genes, proteins, or pathways are overrepresented in a network. Tools like David, GSEA, and Enricher are commonly used for this purpose. These tools compare your network to known biological databases, highlighting significant patterns. Enrichment analysis is a powerful way to connect network structure with biological function, guiding further experiments and interpretation. Statistical analysis is essential for validating network findings. Researchers use statistical tests to determine whether observed patterns are significant or could have arisen by chance. Common approaches include permutation tests, bootstrapping, and false discovery rate correction. Applying rigorous statistics ensures that network analyses are robust and reproducible, increasing confidence in the results. Machine learning is increasingly used to analyze biological networks. Algorithms can classify nodes, predict interactions, and uncover hidden patterns. Techniques like clustering, classification, and network embedding help make sense of large, complex datasets. Machine learning can also integrate multiple types of data, such as genomics and proteomics, providing a more comprehensive view of biological systems. Workx is a powerful Python library for the creation, manipulation, and study of complex networks. It provides tools for constructing networks, calculating metrics, and visualizing results. NetworkX is highly flexible, making it a favorite among computational biologists and data scientists. Its integration with other Python libraries such as Pandas and Matplotlib allows for seamless data analysis and visualization workflow. Graph is another popular library for network analysis, available in both R and Python. It offers efficient algorithms for large networks and supports a wide range of analysis tasks from basic metrics to community detection. eGraph's visualization capabilities are robust, and its performance makes it suitable for handling big biological datasets. Researchers appreciate its versatility and speed. Graph theory provides the mathematical foundation for network analysis. Concepts like nodes, edges, paths, and cycles are central to understanding how networks function. Graph theory also introduces important properties such as connectivity, clustering, and shortest paths. Mastering these fundamentals is essential for anyone working with biological networks, as they underpin all analytical tools and methods. Random graph models are used to generate networks with specific properties for comparison with real biological networks. The Rainy model, for example, creates networks by connecting nodes at random. Comparing biological networks to random models helps researchers determine whether observed features are statistically significant or simply due to chance. These models are a key part of network analysis and hypothesis testing. Any biological networks are scale-free, meaning that a few nodes have many connections while most have few. This property is described by power law degree distribution. Scale-free networks are robust to random failures but vulnerable to targeted attacks on hubs. Understanding this structure helps researchers predict network behavior and design effective interventions. Small world networks are characterized by short path lengths and high clustering. In biology, this means that most nodes can be reached from any other node in just a few steps, and there are many tightly connected groups. Small world properties are common in neural and social networks, contributing to efficient communication and resilience. Recognizing small world structure informs both analysis and interpretation. Biological networks are not static. They change over time in response to internal and external cues. Dynamic network analysis tracks these changes, revealing how interactions evolve during development, disease, or environmental shifts. Tools for dynamic analysis can identify transient connections and temporal patterns, providing a more complete picture of biological processes. Multilayer networks integrate different types of interactions, such as genetic, protein, and metabolic networks, into a single framework. This approach captures the complexity of biological systems more accurately. Analytical tools for multilayer networks can reveal how different layers influence each other, leading to new insights into system behavior and regulation. Robustness analysis examines how networks respond to perturbations, such as node removal or edge disruption. Biological networks are often robust to random failures but sensitive to targeted attacks on key nodes. Understanding robustness helps researchers design interventions that minimize unintended consequences and improve system stability. Resilience is the ability of a network to recover from disturbances. In biological systems, resilience is crucial for maintaining function in the face of stress or damage. Analytical tools can measure resilience by simulating disruptions and tracking recovery. Insights from resilience analysis inform strategies for enhancing system health and preventing collapse. Network alignment compares the structure of different networks to identify similarities and differences. This is useful for comparing networks across species or conditions, revealing conserved modules or unique adaptations. Tools for network alignment use algorithms to match nodes and edges, providing a quantitative basis for comparison. Integrative omics analysis combines data from genomics, transcriptomics, proteomics, and metabolomics to build comprehensive biological networks. Analytical tools for integration can reveal how changes at one level affect the entire system. This holistic approach is essential for understanding complex diseases and identifying multi-level intervention points. Functional annotation tools assign biological meaning to network components. Tools like gene ontology, and KEGG map genes and proteins to known functions and pathways. Annotating networks helps researchers interpret results and generate testable hypotheses, bridging the gap between data and biological insight. Simulation tools model the behavior of biological networks under different conditions. By simulating perturbations or interventions, researchers can predict system responses and design experiments. Tools like Cell Designer and COPASI are widely used for simulating biochemical networks, providing valuable insights into dynamic processes. Several databases provide curated biological network data. String focuses on protein-protein interactions, while BioGrid covers genetic and protein interactions. KEGG offers pathway maps and functional annotations. Accessing these databases ensures that analyses are based on high-quality, up-to-date information, supporting robust research. Despite powerful tools, network analysis faces challenges. Data can be incomplete or noisy, and biological systems are inherently complex. Choosing the right analytical approach requires careful consideration of data quality, network type, and research goals. Ongoing development of new tools and methods aims to address these challenges and improve analysis accuracy. To get the most from network analysis, researchers should follow best practices. This includes validating data sources, using appropriate statistical methods, and cross-checking results with experimental data. Documentation and reproducibility are also crucial, ensuring that analyses can be verified and built upon by others. Let's look at a case study involving disease networks. By mapping the interactions between genes associated with a particular disease, researchers can identify key drivers and potential drug targets. Analytical tools help prioritize candidates for further study and reveal connections that might not be obvious from traditional analyses. This approach has led to breakthroughs in understanding complex diseases like cancer and Alzheimer's. Microbial communities, such as those in the human gut, are complex networks of interacting species. Analytical tools can map these interactions, revealing how microbes cooperate or compete. Understanding microbial networks is essential for studying health, disease, and environmental processes. Network analysis has uncovered new insights into the role of the microbiome in human health. Ecological networks describe the relationships between species in an ecosystem, such as food webs or pollination networks. Analytical tools help ecologists understand how species interact and how changes in one part of the network affect the whole system. This knowledge is vital for conservation and ecosystem management, guiding efforts to protect biodiversity. Network analysis is a rapidly evolving field. Emerging trends include the integration of artificial intelligence, real-time network monitoring, and the use of cloud computing for large-scale analyses. These advances are expanding the scope and power of network analysis, enabling researchers to tackle ever more complex biological questions. Looking ahead, the future of biological network analysis promises even greater integration of data types, more sophisticated analytical tools, and deeper biological insights. As technology advances, researchers will be able to build more accurate and comprehensive models of living systems, driving discoveries in health, disease, and beyond. For those interested in diving deeper, there are many resources available. Online courses, textbooks, and tutorials cover both the theory and practice of network analysis. Community forums and open-source projects provide opportunities to learn from and collaborate with others. Staying engaged with the latest research ensures that your skills and knowledge remain up to date. Let's recap what we've covered. Biological networks are powerful tools for understanding the complexity of life. Analytical tools, from visualization to machine learning, help researchers decode these networks and uncover new insights. By mastering these tools and approaches, scientists can tackle some of the most challenging questions in biology. If you're just starting out, focus on learning the basics of network construction and visualization. Practice with small datasets and experiment with different tools. Don't be afraid to ask questions or seek help from the community. With patience and persistence, you'll soon be able to tackle more complex analyses and make meaningful discoveries. Thank you for joining us on this journey through analytical tools for biological networks. The field is full of exciting opportunities for discovery and innovation. By applying the right tools and approaches, you can contribute to our understanding of life's complexity and help solve some of the biggest challenges in science and medicine. Stay curious and keep exploring.